Okay, next our talk titled RNA-centered strategies for upregulation of beta-catenin in CTNNB1. Both researchers from the National Institute of Chemistry, please welcome Dr. Petra Sušjan and Matea Maruna.
Hello, welcome from us as well in Ljubljana. We are happy to see all the families here because we are dedicating many late nights to this course in the lab, also sacrificing a lot of time with our families. So hope it is all for a successful drug in the end. Okay, so yesterday, we saw some very promising results on the gene replacement therapy, but actually we are working also on at the National Institute of Chemistry at some different strategies, mostly centered around RNA, in order to upregulate the beta-catenin levels in cells of patients with this syndrome. And so just as an overview, I'd like to point out that the times appear to be very hopeful for the gene therapy of rare diseases in general. Just on the diagnostic front, just the development in the DNA sequencing in the last years has allowed to obtain a precise and fast genetic diagnosis. And the decades of basic research are finally paying off. I mean, many protein structures are resolved, cell signaling pathways are well researched, and this really helps understand the disease mechanisms. And just the toolbox of gene therapy strategies is rapidly expanding. And here we actually, for the CTNNB1 syndrome, we are testing many strategies, which I will mention briefly. The rise of parent initiatives, I mean, you are the ones who advocate the unmet medical needs. You're the ones who create campaigns to gather funding for research. And ultimately, this, as has been shown, this creates a great availability of animal models of disease and patient-derived iPS cells, which are helpful to test our strategies in. And also, there's a noticeable reshifting of medical science attention towards fast translation from bench to bedside. And actually, in our institute, we are also working towards establishing a center of gene and cell technologies ourselves, which will hopefully contribute to faster translation of our strategies. And also on the front of the drug approval, there seem to be programs for expedited approval in cases of serious rare diseases. There's, for example, what was mentioned yesterday, the hospital exemption programs, the PRIME program, and compassionate use. And this all results in faster approvals of drugs. Just one exaggerated case was Milasen, an antisense oligonucleotide, which was actually from diagnosis to approval for the N-of-1 clinical trial was actually 1 year. This was by Timothy Wu in Boston. So in CTNNB syndrome, many of these categories seems to be met. So it seems so far that there's this monogenic disease with patients having a mutation in a single gene in CTNNB1. The protein beta-catenin is very well researched, have incredible parent initiatives taking place, availability of animal models, iPSC cells, and also there is already communication with the regulatory bodies that is necessary for the drug approval in the end. So just to say what exactly is the disease mechanism in CTNNB1 syndrome. So in healthy cells, CTNNB1 gene encodes beta-catenin, and this gene is important for cell-to-cell adhesion, that our cells actually stick together better through interaction with cadherins, and for the upregulation of YNT target genes, very important genes in neurodevelopment. However, in the syndrome, loss of function mutations occur in one allele that lead to half of beta-catenin being dysfunctional, not being able to support these functions. And since the gene is haploinsufficient, as we say, this then leads to a spectrum of cognitive and motoric impairments known as this syndrome. And basically our main therapeutical focus is to compensate for the loss of activity from the mutated allele, while ideally maintaining as much as possible the natural gene regulation. So what are the strategies for gene upregulation in haploinsufficiency-related disorders? So you can, as we saw yesterday, introduce a functional new gene. You can correct the mutation through multiple gene editing techniques, or, and this is a unique opportunity in haploinsufficiency, you can upregulate the functional allele from the wild-type copy of the gene. So this can be done either through inhibition of some processes that in the cells endogenously are involved in negative regulation of the protein. You can upregulate the transcription. There are some nice short duplexes called small activating RNAs that we are also working on that seems to bind to the promoters of the gene and upregulate the genes in this fashion. Antisense oligonucleotides then can be used to stabilize RNA, therefore make it be stick around for longer. You can upregulate the translation with ASOs, and another quite innovative technique, you can actually replace mutated alleles in a transcript, and we'll talk about this briefly as well. So just to recap from the first talk today, so the DNA gives rise to an RNA, to a transcript, and this then encodes the protein. But this RNA as an intermediate also undergoes heavy processing. Importantly, the non-coding parts of the gene needs to be spliced out through the action of the heavy machinery called spliceosome, which basically binds to the intron-exon junctions or some motifs that enhance or silence splicing, and they allow for the coding parts of the gene to come together into a mature messenger RNA, and they can come together in different combinations. Some of the exons can be skipped, and that basically provides richness of proteins within our cells. So a very versatile tool that we can employ to modify RNA are antisense oligonucleotides. They're basically short strands of chemically modified DNA or RNA, and the chemical modifications are introduced to ensure their stability, so they are not cleaved by the cell's nucleases. On the other hand, they also influence the mechanism of action of the of ASOs, and then basically bind to a complementary region on the target RNA and alter its processing and expression in various ways. The advantages of ASOs is they introduce a transient modification of the transcript. They maintain the endogenous gene regulation. They are titratable, so it's easier to determine the final efficacious dose. And they don't need any particular carriers. They can just be delivered naked, basically. So they have various mechanisms of action. They can either bind to RNA and attract some enzymes that cause its degradation, resulting in downregulation of the protein, if that is in our therapeutical interest. They can sterically hinder binding of some regulatory proteins involved with translation or some regulatory RNAs like microRNAs, or, and this is interesting for our results, they can modulate splicing. So basically they cover those junctions between introns and exons that I mentioned earlier, artificially inducing the spliceosome to skip an exon that we want to be skipped. And this results in an altered, most often truncated protein with new therapeutically relevant characteristics. And so on this slide, the point of this slide is just to show that several ASOs are already clinically approved or in trials. So this is not such a futuristic strategy as some of the others in our toolbox. The very first ASO to be approved was Fomivirsan, which was relevant for patients with AIDS because due to the immunocompromised system, they actually developed retinitis, and the ASO was injected into intravitreally, so in through their eye, and was quite efficient. But the ASOs that are most important to us are, as a study examples, are ASOs important for neurodevelopmental disorders such as Duchenne muscular dystrophy. So this disease, in a way that is very similar to CTNNB1 syndrome, that it has deletions and mutations are scattered across the entire gene. Therefore, not just one ASO will be effective, because you basically need to, because the way they function is they skip an ASO, skip an exon bearing a mutation and the neighboring exons in order to restore the reading frame of the sequence and provide a protein that is partially active and can result in milder form of the disease. And then important example was also nusinersen, which was targeted at spinal muscular atrophy. This one was interesting because it was universal for all patients, regardless of the type and location of the mutation, because it basically worked by upregulating a gene that was otherwise silenced, exactly because of some splicing modulation. And then a very interesting one was Milasan in 2019, which was targeted at a form of Batten's disease. This is a dysfunction of lysosomes, so cells cannot get rid of their waste properly. And it was interesting about this one, it entered an N-of-1 clinical trial. So basically it was designed for one little girl called Mila specifically. And they couldn't really figure out, despite her having been diagnosed with this disease, what is the genetic cause. And until they saw that actually a retrotransposon has inserted itself into the intron of her, of the lysosomal channel gene, and basically caused the inactivation of the gene. And so once they were able to block the splicing silencing that this inclusion caused, they were able to prolong her life for a couple of years, although the disease was so advanced at this point that she demised at the end. And there are many ongoing clinical trials for ASOs, specifically for Dravet syndrome. It's an interesting one because with the ASOs, they're trying to upregulate the splice variants that would otherwise be non-productive. And hopefully there will soon be one for the CTNNB1 syndrome at the end of this era. So in our group, we're trying to explore the potential of ASO-based strategies for CTNNB1 syndrome. Particularly, first we try to identify what is an opportunity. So I've shown this schematic before, but I didn't say that in addition to beta-catenin being important in these two pathways, it also actually, the majority of it goes into degradation. It is bound by the so-called destruction complex, which consists of scaffold proteins, APC and AXIN, and they trap beta-catenin by binding it, and they also attract various enzymes like CK1 alpha, GCK3 beta, and the ligase, which introduce post-translational modifications. They phosphorylate, they ubiquitinate the beta-catenin, thus labeling it for degradation. And we believe this is an opportunity to therapeutically target, for therapeutical targeting with ASOs. And our strategy is basically to induce skipping of exons that contain motifs important for beta-catenin degradation, because we believe this would result in the beta-catenin stabilization and hopefully retention of its functions. And we actually tested many ASOs targeting different components of the destruction complex. But in the end, it turned out that the best candidate is actually CTNNB1 gene itself, because it conveniently contains the sites important for phosphorylation and ubiquitination, so the degradation motifs, in one easily spliced out exon. And so the question now was to test whether deleting this exon actually really retains the beta-catenin functions. So we, before going into ASO design, we tested the construct where we artificially, which did not bear exon 3, and we found that it had the same YNT signaling properties as the wild-type beta-catenin, and at the same time we also realized that an eteplirsen type of intervention where we would be skipping stop exons with stop codons will most likely not be an appropriate therapy in the CTNNB1 syndrome, because it is the protein is so evolutionally conserved that any disruption leads to a non-functional protein. We also clearly saw that the protein with bearing deletion of exon 3 is expressed. And we also saw that the other function of beta-catenin, namely its interaction with cadherin, is retained, and it is comparable to wild type. And just out of curiosity, how we do this? So basically, we have something called split fluorescent proteins, and we take one half of the fluorescent protein, add it to the one partner in the interaction, and we add the other half into the other partner. And if the fluorescent protein gives fluorescence, it means these two partners are in close interaction. And we also managed to detect co-localization between these partners with alpha-catenin, an important partner in the cell-to-cell adhesion complexes as well. And so then we designed several ASOs and tested them, and this will be explained by Matea.
So further, I will show you results that we have on in vitro experiments. So after these promising results that Petra presented, we decide further to perform screening to identify ASO that can induce skipping of exon 3 in CTNNB1 gene. So here you can see results for our best ASO. So first we wanted to detect efficient exon skipping on mRNA level, and we use qPCR. And so here on this first slide, you can see that our ASO induces sexually exon skipping, and it was around 70%. Then, as Petra already mentioned, beta-catenin is part of YNT signaling pathway. So next we tested ASO if it can upregulate transcription activity of beta-catenin. So here on this slide, you can see that ASO after exon skipping induced transcription activity of beta-catenin. Next, we also checked expression of YNT target genes. And here we successfully detected increased expression for 4 target genes that are part of YNT signaling pathway. Next, we wanted to test this ASO in more relevant cells, and then we decided to test it in a human neuronal cell line. And here we detected same effect. So first we showed nice exon skipping on mRNA level. It was at highest concentration of ASO, it was around 70%. Also, we checked the total beta-catenin expression with ELISA, and here you can also see that we detected increased total beta-catenin expression, and it was like twofold change in comparison to non-treated cells. At the end, we also checked again transcription activity of beta-catenin after exon skipping, and here you can also see significant increase in transcriptional activity. So next step was to test this also in more relevant models like organoids or mouse model. So before we did that, we also tested also in induced pluripotent stem cells with CTNNB1 syndrome that usually produce lower amount of beta-catenin. So far, we detected here again, successfully exon skipping on mRNA level and also increased beta-catenin expression with ELISA. So now, after these promising results, Petra will present further results on organoids and in vivo experiments.
Yeah, so at this stage, we actually contacted our colleagues in Australia, Andrea particularly, who had at this point already developed the CTNNB1 cortical organoids for her gene replacement therapy. And she kindly tested our ASO. We show here some preliminary findings. We saw that the labeled ASO, so that it was homogeneously distributed around the organelles or organoids, and we were able also to show the same skipping as we were observing in our cell models. Basically here we were using passive internalization of ASO called gymnosis, so we did not use any type of transfection reagents, and this is relevant then also for the in vivo applications. And then lastly, we also wanted to check whether ASO upregulates YNT function in mice models. So we are showing just the preliminary results in wild-type mice, where our colleague, whom you remember from yesterday, Dr. Duško Lajenšček, injected our ASO into mice brains with stereotactical injection, together with a reporter of YNT activity, and we were able to observe robust bioluminescence after adding luciferin as well. So this basically indicates that the antisense oligonucleotide type of therapy is quite prospective in treating CTNNB1 syndrome. And it seems that the haploinsufficiency of this gene and the fact that beta-catenin has this degradation mechanism provide a unique opportunity that can be exploited with ASOs. And our designed ASO has shown promising upregulation of the gene in both patient-derived iPS cells and in mice. And an important thing to note is that this ASO-based therapy has potential to be universally applicable to all patients, regardless of the mutation type and location, simply because we are targeting the wild-type copy of the gene that every child with this syndrome has. And this is intended as a complement. This is intended to complement the treatment options. For example, it might be beneficial in patients with less severe disease phenotypes that would not choose the gene replacement therapy. But in our lab, we're also focused into the future. I mentioned there are some futuristic strategies that could at some point maybe be translated to CTNNB1 syndrome use. And one of them is RNA transplicing. So RNA transplicing basically relies on a long RNA molecule that does not only bind to the transcript and induce some sort of splicing modulation, but it brings along exons without the mutation. So this in a way means that we would be correcting the mutation on a transcriptional level. Again, this would be a transient modification. We would not be disturbing the endogenous gene regulation. And importantly, there is theoretically no limit in the size of the exons that we are introducing, so it could be useful in also long genes. It's particularly a problem in Duchenne muscular dystrophy. But of course, the challenge is that prohibit its use, its relevance in vivo right now are currently poor efficiency in vivo. Our main focus here is introducing novel modifications to our trans-RNA in order to increase its transplicing efficiency. And just to show you a snippet of our research to not take too much time. So we are basically using the introns of the CTNNB1 gene to test these modifications. And basically we were able to find that combining trans-RNA, which brings the corrected exons, together with short RNA type of ASOs that inhibit the normal splicing of the gene, actually leads to a significant upregulation of transplicing on both protein level and the RNA level. And with this, I would like to present our team. So in addition to us, Dr. Duško Lanšček, Professor Dr. Roman Jarala, I'd like to thank kindly Professor Dr. Leszek Lisowski and Dr. Andrea Perez Iturralde for trusting our ASO enough to dedicate some of their time to test it in organoids. And of course, to Dr. Špela Mirošević. We consider her a fellow biomedical researcher, biochemist, and only yearly in these yearly conferences, we're actually reminded that she just wanted to be a parent. So thank you.